Our last speaker of the session is Pamela Anderson. Pamela Silver.
You know, if you Google me.
Oh boy. Okay. Well, there you have it.
All right, the session's over.
Pamela Silver.
Now I know. That's what you think of me.
Okay. And I'll just let her take it from there.
I don't think I've ever had an introduction quite like that. Thank you, Christina. I'll have to come back with that one. My feeling was that microbes have really been the powerhouse for synthetic biology, and a lot of the early experiments and the ability to predict how systems behave has been carried out very elegantly by some of the people in this room in microbes, and in fact, E. coli may in fact be the well-oiled machine. So I'm going to talk to you about our attempts to move beyond microbes, give just a brief overview of, or brief review of some experiments we did in yeast that I've actually presented here last year, and then tell you about how we've translated that system into mammalian cells. So this is a brief summary. I'm going to talk about how we can start to use parts to build higher eukaryotic cells that can begin to compute and store memory. And I'm going to talk about something that I call post-transcriptional logic. It's a bit of a biology lesson, but it has a synthetic biology component to it. And then I'm going to talk about some interesting experiments that have to do with quantitative protein design. I'm not going to talk about bioenergy. We have lots of posters on this, so see our posters. Okay, so I think this has been one of the overarching themes of this meeting and of synthetic biology. So can we make biology easier or more predictable to design by using parts? I think one of the critical things that we've seen some of here is having really good measurements of the behavior of the system, and then the ability to model the system. And I'll give you a flavor for how we've gone about doing these things. Okay, so first let me begin with cells that can remember, and this first part will be review. So several years ago, we built in yeast a simple device where the cells could remember that they had been exposed to a signal. So very simply put, the idea was that a yeast cell was exposed to a small molecule signal. It turned green. When you removed that signal, it remained green, and that's how that was indicative that it remembered that it had been exposed. So we did this in a very simple way, the way nature would do it. We built a simple positive autofeedback loop from synthetic parts. So these are synthetic transcription factors. They're transcriptional activators. Each of these blocks represents a piece of synthetic of DNA that was synthesized. So the idea here is that A is the small molecules. So when cells are exposed to A, they make a transcription factor that is red. It's an activator. It's targeted to the nucleus. Has a DNA binding domain. The cognate DNA binding site is on a second activator called B here. So A turns on B. B is green. It also has an activator. It's targeted to the nucleus, and it's capable of turning itself on. So we can then remove A, and B can sustain itself. So I'll show you the data very quickly. So we did this first in yeast. So here are yeast. Here's when they have been exposed to nothing, so there's no signal. But when they're exposed to A, we have red nuclei because A is turning on this activator, and in turn, this is turning on B, and so we have green nuclei. However, if we remove A, the stimulus, the red goes away and the green is sustained. I'll show you this in real time because this is relevant to making measurements of the system. What we do is we make single-cell measurements of the behavior of these systems so that we can ultimately develop accurate, predictable models. So I'm going to play you a movie, as I said, and what you're going to see here are the yeast cells growing. The red signal will disappear, but the green signal will be sustained, and you'll see these develop into a colony. If you can't see it, you can see it on YouTube. Great place to post your data, by the way. All right, so hopefully you can see here the cells dividing. The signal is disappearing, and then over many cell divisions, the green signal is sustained. So this is memory in living yeast cells. Okay, so that's kind of where we left the story last year. So then what we did was we moved this system into mammalian cells. It's the same idea. We have two synthetic activators. They are turned on. This one is under control of doxycycline, and then it has a cognate DNA binding site. So again, we have, the colors are reversed here, but this one is green, and the red signal is indicative that the cells have remembered they've been exposed to the small molecule. So these are mammalian cells. This is an overlay. Both proteins are in the nuclei. This is so they're yellow because they have both green and red present. But when we remove the signal, some of the cells sustain the red signal, indicative that they have remembered that they were exposed. Now, this system is not by any means perfect, and it brings up a lot of the problems that come up with working with mammalian cells as opposed to yeast, where you can target things specifically into specific sites in the genome, et cetera. So we added on to this system to make it tunable. We added a repressor to make sure that the memory loop was really off. And then when we turn on the system, within the intron, we have inserted microRNAs. And so these microRNAs then can turn off the repressor so that the memory loop can run. We modeled this system, and I'm not going to go through this in detail, but to show you that we can find a regimen that is just right. So in this case, for example, we would have too much repressor, so the trigger signal would never be high enough to see the memory signal. But we can find a regimen where we can repress the repressor sufficiently so that the trigger is on and you can see the memory. And we've actually been able to carry out experiments, and this is some early data to just give you a sense, and I hope you can see this. In fact, the repressor signal does go down when we add doxycycline, which is what you would predict, and we can also see the memory loop in action. I hope you can see this. I apologize for it being somewhat overexposed, whereas in the absence of doxycycline, the loop is not on. All right, so where do we want to go with this? And so this is where I just want to sort of project into the future what I think we can start to do with mammalian cells. So one of the things we're doing is building a device that will remember exposure to DNA damage. So imagine that you have a system, much like I described, where a cell is exposed to DNA damage. It sends out some kind of signal, it turns color, and then that cell remembers over time that it was exposed to DNA damage. So this is actually a system that we're in the process of building by putting in a p53 responsive promoter into the trigger. Now, what's interesting about such a system, you might ask, well, why do you want to do this? So one thing that would be interesting is that you can, that in principle in a tumor, this would allow you to identify the initiator cells, that is the cells that were initially exposed to DNA damage, and then if those cells could remember as the tumor progressed, you would be able to selectively isolate out that population of cells and study them. And this is a way of getting at the initiator cells from a tumor, which has been one of the major outstanding questions in tumor biology. And then you could take this even one step further and sort of use it to do what nature already does, but do it in an engineered way. So to have these intron-encoded microRNAs downregulate whole pathways in the cell and meanwhile upregulate specific pathways. So we imagine that you could use this to build essentially a synthetic differentiation system. Okay. So, briefly taken together, what we've been able to do is design and characterize a simple system and be able to predict its behavior. This is particularly true in the yeast system, which we published several years ago, and I hope I've given you a flavor that we can start to predict the behavior in mammalian cells as well. The system is composed of interchangeable parts. You can put in different DNA binding domains, different promoters, make it responsive to different stimuli. And I hope I've convinced you that the system is transferable. So what's nice is that we can characterize the system in yeast. We can get a sense of whether it will work or not. Yeast grow much faster. We can manipulate them. And then we can transfer these systems to higher cells. And then in the long term, we imagine that we could start to build cells that would, for example, report on DNA damage. We could also imagine cells that would, for example, deliver drugs in a pulsatile manner in response to signals. Okay, so now I want to move on to something that's part science, part synthetic biology, but I think it has implications for synthetic biology. So as most of you know, there's a basic conundrum in the genome in that there aren't enough genes to account for the complexity of the organisms. And part of the way that the organism deals with this is through alternative splicing. So the genome is laced with introns, and this allows you to create a lot of diversity out of a single gene. However, there's a conundrum in that genes, many of these introns are very long. And I just illustrated some examples here for you. So here you have an average gene length of 27 kilobases, but the mRNA that results on average is about 1.3. And then you have really giant genes like the dystrophin gene, which has many introns and leads to a fairly large size mRNA. So in biology, there are a lot of questions around this problem. How do you get the correct splicing? What's the impact of having all these introns? How do you get these RNAs processed in a reasonable time frame? So let's think about what this might mean. What are the consequences of introns for gene expression? And I promise you I will get to a synthetic biology experiment. So this is not just about splicing, trust me. So if you have a single transcription activator, so if you have a short gene and you're looking at time and appearance of mRNA, you would have a certain curve at which this appears. And a longer late gene, which takes some time to be processed, would be delayed. Similarly, if you had a repressor, then the shorter gene would be repressed quicker than a longer late gene. They had a shared repressor. And then lastly, if you had a case of delayed autoinhibition, you would predict that if you have a short gene, it would give a higher frequency of oscillations as opposed to a longer gene would give a lower frequency. So these are some of the predictions. So and there actually is some interesting biology behind this. Again, this is developmental biology, but there is an interesting example. This is actually a developing zebrafish, and this is observing a specific gene, and Lewis put forth a very interesting model that these differences in expression are due to oscillations in the expression of the gene. Now, the other observation that intrigued us is that many genes involved in early development have a single very long 5' intron. And I'll show you why. And that the idea is that this may somehow contribute to this oscillatory behavior. Okay, so here's the synthetic biology experiment. We built a synthetic gene, again from parts. It's green. It has a degradation tag. It's targeted to the nucleus. And it's capable of repressing itself. And into this, we inserted a cassette so that we could vary the length of the intron that was inserted. So just a big brief review of what's going on here. transcription begins, there's a certain rate of elongation of polymerase as the gene is converted to RNA, during which time the introns are spliced out. The RNA is exported out of the nucleus, converted into protein, and in this case, the protein would return to the nucleus and repress itself. So this is the system that we built. Now I'm going to play you a movie. Where I want to hope that you can see the oscillatory behavior of this system. If you can't see it, just appreciate that these are living cells under a microscope filmed over a very long period of time. The protein is targeted to the nucleus and it's fluorescent. And people tell me that if you focus on this nucleus, you can see the oscillations. It's a little complicated. Your eyes play tricks on you because, remember, these cells are also dividing at the same time. I can play it again, but let's give it a shot. Tell me if you can see this guy. Can you see it oscillating? Yes? Great. Like I said, it's a little complicated because you've got cells dividing also. All right. So if you didn't believe me, this is not the U.S. stock market. It's not that low yet. Maybe Monday's opening. However, this is one of those nuclei now observed over time, and you can see the oscillation, and you can also measure it. And again, I want to enforce that one of the principles behind synthetic biology, I think, that is important is this idea of making careful measurements, and in this case, single-cell measurements, which is what we've done with these mammalian cells. Okay, so what does that mean? So then we varied the intron length. We have introns of 3 different lengths, and I don't expect you to get much out of this except that each of these are traces of the oscillatory behavior of individual cells with different length introns. This is the more meaningful piece of data, at least visually, which is looking at the average period length. And the point here is that as the intron grows longer, the period length also gets longer. All right, so what could that mean? So when do these bursts in expression occur? So they could occur at the initiation of transcription, or they could occur during the period of elongation. So we used mathematical simulations to test those 2 possibilities. So if there are no bursts in transcription, then you actually get perfect periods, as illustrated here. On the other hand, if you create a simulation where you incorporate the bursts into elongation, so elongation is not a continuous phenomena, then you see these differences in the period. And here's the histogram that you get from the mathematical simulation. And when we compare that to the actual experimental data, it's actually quite striking in that the correlation between gene length and the periods of oscillation, if we compare the experimental data to the predicted data, to the simulation data, they look virtually identical. So what this leads us to think in terms of biology is that when you have these longer introns, that these changes in oscillation are due to the unevenness of the elongation and to actually stalling and then bursts in the elongation of the polymerase. Okay, so what does this mean? So we've observed that we can build a delayed autoinhibition system in mammalian cells, and this exhibits pulses of gene expression. The period mode increases with intron length, and I showed you a model where there's some kind of pausing going on, and this could be during splicing. What I think this suggests is that the intron could be a part that you could think about as a timing device. And again, we're interested in whether this could be a general mechanism for very precise pulsatile delivery of a gene product. Okay. So now I want to talk about the third set of experiments that we've done. So I want to move now not only out of the nucleus into proteins, but I'm going to move out of the cells. So I'm going to talk about phenomena happening on the surface of cells. And the underlying premise for these experiments is the following. First of all, a classic paper that I recommend all synthetic biologists read. In fact, all biologists should read, and it's not written in French. But it has a very important conclusion that reduction of dimensionality in biological diffusion processes is a key design feature. And I think that's something we should all keep in mind. The new thing that I'm going to add to the mix here in terms of these experiments is the concept that actually weak binding can be a good thing in biology. All right, so just as a reminder, what kinds of processes do we think about in terms of diffusion in biology? One that I've thought about a lot through my years, having worked in the Patashny lab when a lot of this was being figured out, is the question of how do proteins find their correct binding site on DNA. They have a general non-specific affinity for DNA, and then they can move along DNA and search for their proper higher affinity binding site. Now, you may not have thought about this, but a similar thing happens on the surface of cells. So if you have a ligand for a receptor on the surface of now a mammalian cell, the ligand has a certain non-specific affinity for the membrane bilayer, and simultaneously receptors are actually moving laterally in the bilayer. So there's a lot of two-dimensional diffusion that is coming into play in terms of how ligands find their receptors on the surface of membranes. All right, so what does that have to do with synthetic biology? So I want you to imagine the following problem. Let's take a drug, a small protein drug like interferon. So interferon can be used effectively to kill cancer cells. The problem with interferon is that it also binds to virtually every cell in your body. It has an interferon receptor, and if you've ever taken it or known anyone who has, you generally feel like crap. And so what you would like to do, and I'm just using interferon as an example. I'll get to some how we built this into a more general platform technology in a moment. But what you would like to do is have a way to selectively target something like interferon to, say, a cancer cell. Okay. So imagine that we have a small protein, which we call the activity element. We have a targeting element that will deliver it to the correct cell. These are linked together by a short stretch of amino acids. Now, for the aficionados amongst you, you're saying, well, no big deal here. We know how to make protein-based drugs. Now, what's different here? So imagine if you could reduce the affinity of interferon for its receptor, but then by increasing the concentration, the local concentration on the correct cells, you could take a bad binder and make it into a good binder. And I'll try to illustrate that for you. So what we did was make mutations in the activity element. In this case, interferon was nice because we knew the structure of how it interacts with its receptor, so we could make very specific mutations that we knew would lower the affinity by certain amounts. Okay, so this is an illustration of what I just said. So imagine you have a mutated activity element. It can't bind to its receptor. It binds with lowered affinity. However, if it's delivered to the cell by a specific targeting element, this increases the local concentration so that a bad guy can now bind, as opposed to what would happen on a non-target cell where the binding would be blocked altogether because you've mutated the activity element and there's no targeting element on the cell. Okay, do you get it? People following this, hopefully? Okay, so we did this. So we did a first test case. We did with two proteins we know very well, interferon alpha and EGF. EGF is an interesting test case because tumor cells are increased for EGF receptors, so this has some potential therapeutic value. But as I said, the thing that was particularly attractive about this is that we knew the structures of the proteins. We were able to very accurately make mutations that would reduce the affinity by an order of magnitude of interferon for its receptor, and we were able to actually build a predictive system for how long to make this linker, which I can talk about with anyone who's interested. Now, I don't want to go into how interferon works. I just want to say again, we have an assay. This is not a single cell assay. This is taking bulk cells, and all you have to know is that there's bands here. These come from a gel. This is indicative of if there's a band here, then the interferon is active. And so the important comparison to make here is, first of all, in this case, so this is the mutated interferon fused to EGF. There's a band here. It's active. If you compare it to the mutant interferon alone, there's no activity. So the experiment basically worked. So that was kind of exciting. We took it one step further, and this gets a little technical for some of the cancer aficionados amongst you. Cancer cells make a specific form of EGF receptor for which there's a specific antibody. So instead of using EGF, we use this specific antibody. We created the same fusion. This special kind of mutation in the EGF receptor is common in glioblastoma. And this experiment came out very nicely. Now we're comparing a glioblastoma cell line, and here's the monoclonal antibody fused to interferon. And you can see that it's active here. And again, the important comparison to make is between the mutated form and the form that is specifically targeted to the cells. And we've quantitated this data, and it's absolutely amazing. This is several orders of magnitude difference. So I think this one actually may be ready. This is going into mice for animal experiments, and so we're quite excited about that. I left out one slide here. Sorry. I'm not going to talk about bioenergy, but I left out a slide that concludes what I wanted to say about these proteins. I think that they indicate that we can build a platform technology around this idea. And so we've done this with things beyond cancer. And one that we're particularly excited about is with EPO. So again, you have a situation with EPO, very commonly used small protein drug, used in cancer patients to treat anemia that is due to chemotherapy. There's a problem in that recently data has come out that shows that EPO stimulates the growth of tumors, so it's almost like giving growth serum to the tumor. So even though it makes you feel better, it doesn't prolong your life. So again, you'd like to have a situation where EPO is specifically targeted to red cell precursors. Again, we know the structure, so we've been able to make a mutant form of EPO and target it specifically to red cell precursors. Works like a shot. So we're very excited about that. Lastly, we want to save the planet through bioenergy. And I'm not going to talk about this, but this is just a teaser for our posters. We want to convert sun into energy. We imagine a microbial solar cell. And I'm going to come back to this at the end and let it play. But first, I want to acknowledge some of the folks who did the work from my lab here. Some are at this meeting. Caroline, in fact, many of them are at this meeting. Bruno, Dave Savage. This is us in a 12-meter racing the Endy Lab. And you might ask yourselves, where's the Endy Lab boat? They're way behind. Take that, Drew. And then just as a teaser, I'm going to let this movie run. These are, just because I think it's cool, it's almost like art, and you can ask me what it is later, and you can also ask me questions. Thank you. Yeah.
I have two questions. The first question about the first part is I have not found your modeling, especially circuit. More detail is I want to how do you choose the parameter to fit in your experiment. Another question is about.
Why don't I answer the first one because I'll forget what it was. Okay, so it was how to choose the parameters for the modeling. So when we modeled the yeast work, if you read the paper, that we actually used measurement data. So we very carefully measured the in vivo KD of the activators for their promoters and the rate at which they activate, et cetera. So we made a number of very careful in vivo single-cell measurements, and then we used that data to fit to the model. And so we're trying to do a similar thing with the mammalian cells as well. So we're really going more from the data to the modeling. And your second question was?
The second question, I can't follow you about the two receptor, how they can organize on the memory. I mean, our two receptor is couple or not? If not,
I have
why
a feeling I didn't explain something clearly. I'm not understanding your question.
I mean, why if you engineer the two protein binding target and they can improve a mutant protein to find its receptor on the memory.
Are you understanding the question?
Sorry.
Are you understanding the question?
Somebody was asking how the two receptors on the membrane localize. So I think he's maybe getting confused. He's wondering, this time I thought it was, how the two proteins that you're linking give better binding when one is mutated. I think
Okay, but that's the whole idea. So the idea is that by locally concentrating a bad binder, you can turn it into a good binder. So that was the kind of the idea of the experiment. I don't know if I'm answering your question or not.
Pamela?
Yeah. Uh-oh.
A different question. Which human cells did you use? Was this stem cells or something?
No, I can go back, but so in the second experiment, that's a glioblastoma cell line. So we used all cell lines. So and in the case of the proof,
glioblastoma is brain tumor.
yeah, but they're cell lines. They're not primaries. And in the EGF experiments, we used a lot of different cell lines. The one that was probably the most interesting were Dowdy cells because they don't have EGF receptor, and so then we transformed them with EGF receptor. So we had a nice control cell. We had a perfectly matched cell lines, ones with and without EGF receptor.
They were very nice experiments, just like Lambda.
Everything's like Lambda.
Yeah.
There's everything in the world.
Well, you came from Mars.
Yeah, everything is Lambda. You know, so Lambda's perfect. Yeah.
Hi,
Now
Pamela.
that we know Lambda's perfect, yeah.
I was just wondering if you would comment on whether you thought the difficulty in scaling up from going from yeast to mammalian cells, was that more of a challenge than you expected, or did it turn out to be more simple than you
Compared
expected?
to other things I've done in my life, I didn't think it was that challenging. But not to be facetious, I think that, you know, the thing we struggle with mammalian cells, and there's many people in the audience here who can elaborate on this as well, We don't have the luxury we have in yeast. This is one of the many things. We don't have the luxury, with some exceptions, of being able to specifically target genes into the chromosome. So a lot of this is done randomly, and then we sort for the cells that give us the right behavior. That's not necessarily hard or a bad thing to do. It's time-consuming. And then you don't know that much. I mean, you could analyze the locus where it's inserted. The nice thing in yeast is you know, you can say, okay, I'm targeting it to the URA3 locus, and I know a whole lot about how that behaves. So I think we will be there with mammalian cells someday, but so I'll just say that's one problem. They grow slower. They do weird things. Any other? Yeah. Oh, okay. A little off topic here. This is back to microbes. So just for fun, this is a little advertisement for Dave Savage's poster. So these are cyanobacteria. So they're a photosynthetic bacteria. They're actually green. But what we've done is label it's a structure inside the cell called the carboxysome, and we've tagged it with GFP because we're interested in how the carboxysome fixes CO2. And so this is the first, to my knowledge, visualization of carboxysomes in living cells. So that's what this is. It's red, fluorescing red because they're green. They're photosynthetic.
Okay, so there was a question there.
Thank you. I looked through several topics concerning synthetic biology and found not so many ideas concerning eukaryote systems. So I want to ask that what advantage do you consider that in the eukaryote systems besides its applying potential?
What is the potential in
applying potential?
There's a million ways to answer that question. And by the way, as an advertisement, I think there's
available in the near future.
That's very interesting. And I see Ron sitting back there, right? Your student is talking next. Where is she? Anyway, there's going to be a very interesting application in the next session, which I'm running to advertise. So let me just answer your question two ways. First of all, eukaryotes do more things, and I hope to illustrate that with the Intron experiment. So you have more parts to work with. You have different behaviors. You have microRNAs. So I hoped to give you a flavor of the increased, you can take advantage of the increased complexity to build systems that you have more parts to work with. Secondly, from a medical application point of view, there you can start thinking about building cells that will differentiate in specific ways, cells that will secrete drugs or small proteins. I could go on forever. I mean, there's a million ways to answer this question.
Yes, and I think of another thing as that the eukaryote system has another advantage that often they can interact with each other, that they can form a community, and through proliferation and differentiation, they can cooperate to have some new functions. So, do you think that's also a way to a direction of research in the near future?
Sure. I mean, one thing we're trying to do is, and I'm sure other people are thinking about this too, is build systems where you can program in asymmetric cell division, which is something that happens during development, and yes. It would be that's an example of something that higher cells do that would be very interesting to program.
All right, one more question.
It was a really nice talk. I was sort of interested if you could comment on the periodicity you saw in your oscillators. It looked like the period was increasing over the course of that trace, and then maybe also commenting on where you think the source of the wide distributions is. It seemed like the distribution of periodicity for a given intron length was wide.
So the distribution gets wider as the intron length grows, okay? Or the period gets longer.
Longer, but also it seemed like the distributions were awfully wide given an intron length.
So it gets more, you're right. So I didn't really emphasize that. It does get more erratic as the intron gets longer. Is that what you're commenting on?
Yeah, or even just even for the shorter introns, it seemed like over the course of the experiment, for each each cycle of the oscillation, the period was increasing. So it wasn't a constant period oscillation.
That is, I'm trying to think if that's a true statement or not. Okay, let's say it is. Okay, so what's the question? Why? I don't know
any
why.
ideas what the sources might be, but
I don't know why. I think we only went as far as trying to figure out the source of the oscillation as being connected to elongation during transcription. But I think your question is well-founded because I think there's a lot more you could do with this system than what we've done with it, and I think it's a, if anyone wants to work on it, it's a really interesting system.
All right. So with that, if there are other questions for the speakers, please find them after the end of this. And thank you.